So this is the video on the proteins chapter, and so we're going to talk a little bit about proteins. You might recognize this particular complex here from the video that we watched on the first day. This is the ribosome, so this is the small ribosomal subunit, the large ribosomal subunit. This is the RNA being read, the triplet code being read and translated into the growing polypeptide chain that's coming out here from the um, from the large ribosomal subunit. So some of the things that I got out of the chapter, one is that proteins are a polymer. They are amino acids linked together by peptide bonds. That's a dehydration synthesis. And so since it's an anabolic reaction, it's going to be requiring that we put in energy. So that after those amino acids are linked together by those peptide bonds, there's going to be a sequence of amino acids that we call the primary structure. And then self-assembly happens to that primary structure just based on what the primary structure is, that they will create conformations that have some amount of stereotypic nature to them. There are a couple that recur over and over in proteins. Those include the alpha helix, the beta sheets, helix loop helix, and some others. There may be um, some unstructured regions in between these primary structure motifs or secondary structure motifs, but there are these secondary structures, conformations that are recurrent, happen all the time. And so we call that the secondary structure. The many secondary structures coming together into a tertiary structure that recurs from protein to protein to protein are called domains. And so these might be um, a a portion of a protein that always attaches to this other domain. And so proteins can use these kind of like Legos or Tinker Toys to create structures that interactions with other proteins and stuff like that, or catalytic domains that recur from protein to protein to protein. So then you start to have some amount of tertiary structure. The tertiary structure is the overall 3D structure of the protein. So when these domains come together, uh, then you get your globular protein, say, that would be the tertiary structure. A quaternary structure is when more than one gene product or more than one individual protein come together to make the functional unit. We call that the quaternary structure. So proteins are the result of the central dogma of biology. The so-called central dogma of biology um, is that there's information in the DNA, and that information gets transcribed into a message RNA, and then that message RNA gets translated by a ribosome into the primary structure of the protein. So that's the central dogma of biology. There are some exceptions to that. There are RNAs that never get translated, or viruses that can take their RNAs and insert um, things into your DNA, or RNAs that make DNA out of them that then gets transcribed. So there are some exceptions to the central dogma of biology, but for the most part, the way that proteins in your body are made is the DNA to RNA to proteins model. And there are lots of proteins that can be made. This is really how things happen in your body. DNA doesn't do anything; it just sits in the nucleus. It's not going to be able to carry out the functions of life. RNA, on the other hand, there are structural RNAs, there are catalytic RNAs, there are informational RNAs. So RNA could do it, but we've refined that from the earliest time. There were only RNAs, but we've refined that to create DNAs and proteins. And so in your cells, the most functions get carried out by proteins. And there are all kinds of different proteins that get made. There are structural proteins, like, for instance, the microtubules. The microtubules are made out of tubulin dimers and create this structure of the cell, as well as a superhighway system that the motor proteins can walk down. There are communication uh, proteins, cell adhesion molecules that can convey information from one cell to another, or give some structure where these cell adhesion molecules may bind two cells together or a cell to the extracellular matrix. There are proteins on the outside of the cell, the extracellular matrix. There are motor proteins that create motion inside of the cell. The kinesins, the dyneins, the myosins walking down the cytoskeletal elements to deliver things inside of the cell. Um, there are catalytic proteins. So here we see an actin fiber being cut by an enzyme. Maybe there's some sort of reorganization of the actin cytoskeleton here, because you can see back here there's actin cytoskeleton being held together by some linker proteins, and so some reorganization could be capable if we had an enzyme that could cut actin. So there are catalytic proteins. So there are all kinds of different proteins, and all proteins pretty much have this primary, secondary, and tertiary organization. Some have quaternary organization, some do not. So what is primary, secondary, and tertiary organization? I've alluded to this already, but the primary structure is just the left to right reading of the amino acids in sequence. And so this is the structure that is coded by the DNA. The A's, C's, T's, and G's of the DNA is transcribed into an RNA, and then that triplet code is read by the ribosomes to decide which amino acids to put into the protein. And so the left to right reading of the amino acids, glycine, alanine, serine, asparagine, phenylalanine, valine, tyrosine, cysteine, etc. This reading is the primary structure. You have amino acids. Uh, Different amino acids have different R groups, but they all have an amino on one side and a carboxylic acid on the other side. Then through a dehydration synthesis, that carboxylic acid will be joined to the amino on another amino acid. So we'll take away a water, take away an OH off of the carboxylic acid, the C terminus of one amino acid, and a hydrogen off of the amino group, the N terminus of another amino acid. Taking away that water causes a bond, a peptide bond, which is a kind of covalent bond. And we'll just do that over and over and over inside of the ribosome to get our primary structure. In this case, methionine, aspartic acid, leucine, tyrosine. So methionine and leucine are both nonpolar, and aspartic acid and tyrosine are both polar. So nonpolar amino acids are going to be hydrophobic and not want to be in an aqueous environment, which most of the cell is an aqueous environment. And so they're going to have to sequester themselves away on the inside of the protein. The polar amino acids, on the other hand, can interact with the water milieu of the cytosol or the interior of the um, individual vacuoles or organelles that they're inside of, and so can stay in solution. And so every amino acid has some amount of hydrophobicity or hydrophilicity. And so the more charged something is, like arginine and lysine, the more hydrophilic it's going to be. The more long hydrocarbon chains it has, like isoleucine, valine, leucine, the more greasy it's going to be. And so the more hydrophobic it's going to be because oil and water don't mix. And so, for the most part, the hydrophobic amino acids are going to have to be sequestered on the inside of the protein, or maybe they're going to be the part of a protein that is going to be able to span the phospholipid bilayer and be integral membrane proteins. So, moving on from primary structure, we start to see the secondary structure. The secondary structure is achieved by hydrogen bonding within the backbone. The backbone was this line going along here of HNCC, NCC, NCC, NCC. That's the backbone, and there's going to be a series of hydrogen bonds within the backbone between different amino acids. And that's going to cause the secondary structures. There are two depicted here, the alpha helix, which is 3.6 amino acids per turn, or the beta sheets. Beta sheets can run in parallel, or in this case that you see here, anti-parallel. And that just has to do with the N-terminus, C-terminus order of the backbone. But alpha helices and beta sheets are the most common secondary structures. Alpha helices are the most common secondary structures for membrane spanning proteins. Um, in addition to alpha helices and beta sheets, we are going to start seeing other interactions, and those other interactions are going to allow for the tertiary structure or the overall globular protein nature of the protein. And these can include hydrogen bonding, again between the side chains, or hydrogen bonding again within the backbone. So either between the backbone O's and H's or N's and H's, or between two R groups. We'll also see some ionic bonding between R groups, where you have a negative R group and a positive R group are going to be able to interact with each other. We are going to see some van der Waals forces. That's what I was talking about before, where the hydrophobic portions try to sequester themselves on the inside of the protein. And then there's also these disulfide bonds between cysteines, and those are for after the globular nature of the protein is established, to lock it in and to stabilize that structure. So the disulfide bonds give an overall rigidity to the structure. And then these tertiary structures, they can be all kinds of different things. Like you can have a lot of alpha helices interacting with each other, or a lot of beta sheets interacting with each other. And then quaternary structure, the last one, is more than one protein interacting. Hemoglobin is a protein that is pretty well known, and hemoglobin is made up of two hemoglobin A's and two hemoglobin B's. So I guess now we'll move on to the second video.